So one of our new tiles is Salmonella. It's time to start exploring what our immune system does when we have an actual bacterial infection in the gut, and Salmonella is one of our more common bacterial infections. So here we are under Salmonella poisoning. By the way, today when I looked it up using the search box, it came right up, so sometimes it works. You could also look at Salmonellosis and the species Salmonella enterica. And of course, we've got the usual videos. So what are the syndromes that result from Salmonella poisoning? Gastroenteritis. You guys are probably familiar with how food poisoning feels a lot like the stomach flu. That's gastroenteritis. It can also cause enteric fever, which is another name for typhoid fever. It can cause bacteremia, with or without metastatic disease. Salmonella can get in our blood. Usually our immune system's going to take care of it, but some people have a hard time handling it, especially people with sickle cell disease or any other disease that causes an iron overload. So if you know you have an iron overload, beware of the problem with Salmonella. It can get into the blood. Once it gets in the blood, it can settle in the bone. That's called Salmonella osteomyelitis. It can also have a tropism, which means it homes in on the circulatory system where it's headed for atherosclerotic plaques, and that could cause a mycotic aneurysm of the aorta. The term mycotic's misleading because it refers to fungal, but in this case, it just means an infection. So, if you're over 50 and you get Salmonella, it can get into the blood and settle in plaque and cause an aneurysm. There can also be asymptomatic carrier states. Non-typhoidal Salmonella is widely disseminated in nature, and it's intimately associated with animals. It contaminates half of the chickens in the U.S., which is why they dip commercial chickens in bleach before they head to the market. It's also in eggs, unpasteurized milk, fruits and vegetables, marijuana, and reptiles. Typhoid fever is caused by a certain type of Salmonella called Salmonella enterica serotype typhi, or just Salmonella typhi. The gallbladder is the organ that harbors Salmonella. One of the major risk factors is eating out in a fast food restaurant. Bacteremia in salmonellosis is usually transient and inconsequential, except for those conditions I already mentioned, and also underlying heart disease, endocarditis. In young children, it can be a factor in meningitis. Okay, let's talk about IBS. I've mentioned this in previous classes. There is a potential link between a Salmonella infection and the development of irritable bowel syndrome. Remember, that's different from irritable bowel disease. Irritable bowel syndrome is a chronic functional gastrointestinal disorder characterized by symptoms like abdominal pain, bloating, diarrhea, and constipation. And this connection is explored in the context of something called post-infectious IBS. That's a subtype of IBS that develops following an acute gastrointestinal infection. Here we're going to talk a little bit about the evidence and the mechanisms behind this. So there is epidemiological evidence. Studies have shown that a significant percentage of those who develop IBS-like symptoms after recovering from bacterial gastroenteritis caused by pathogens like Salmonella, also Campylobacter or Shigella. Research indicates that the risk increases with the severity and duration of the initial infection. So Salmonella infections that cause prolonged inflammation and diarrhea are particularly implicated. As far as the mechanisms go, several mechanisms have been proposed. For example, the persistent inflammation. Even after the infection's cleared, Low-grade inflammation can persist in the gut because Salmonella can damage the intestinal lining and activate immune responses that don't fully resolve, leading to a heightened sensitivity and altered gut function. Studies have found increased levels of inflammatory markers, in other words, cytokines, in the gut mucosa of post-infectious IBS patients. Another mechanism at play could be gut microbiota disruption, because Salmonella can disrupt the balance of the gut microbiome. As we know, that's a key factor in digestion and immune regulations. So this dysbiosis, the imbalance of gut bacteria, that can persist after the infection, contributing to ongoing symptoms like diarrhea or constipation, hallmarks of IBS. Another factor could be altered gut motility. So the infection can affect our enteric nervous system. Think of that as our gut-brain connection, leading to changes in intestinal motility. Salmonella toxins and inflammation can damage nerve endings in the gut, resulting in abnormal contractions and hypersensitivity. And of course, increased intestinal permeability. It can compromise the integrity of the intestinal barrier, think leaky gut, allowing bacteria or toxins to pass into the bloodstream more easily. And then there are also psychological factors. Severe infections can trigger psychological stress, for example, anxiety about food or health, and that can amplify gut-brain axis dysfunction. As far as studies go, I'm going to skip this part, but they're here if you want to look at them. What about risk factors? Not everyone who contracts Salmonella develops IBS. Risk factors seem to be severity of the initial infection, where we see prolonged diarrhea or even hospitalization. And women are more prone to IBS in general. Also, a younger age at the time of infection, and pre-existing psychological conditions like anxiety or depression. As far as clinical implications, the link between Salmonella and IBS highlights the importance of preventing foodborne infections through proper hygiene and food safety. Also, monitoring patients after a severe gastroenteritis episode for persistent symptoms, and exploring treatments like probiotics, anti-inflammatory agents, or dietary interventions to restore gut health after the infection. Now, what about irritable bowel disease? And that's going to be Crohn's or ulcerative colitis. Inflammatory bowel disease involves chronic inflammation of the gut, driven by an interplay of genetic predisposition. Now, they say genes are very important in terms of whether or not it's going to move into irritable bowel disease. Immune dysregulation, environmental factors, and the gut microbiome. While Salmonella infections are not considered a primary cause of irritable bowel disease, they can act as an environmental trigger or exacerbating factor in genetically susceptible people. And we've got epidemiological evidence. I'm going to skip through that. You guys can come and read that. As far as the mechanisms go, immune dysregulation. Salmonella is a pathogenic bacteria that invades the intestinal mucosa and triggers a robust immune response involving pro-inflammatory cytokines like interleukin-6 and tumor necrosis factor. In genetically susceptible people, this acute inflammatory response may fail to resolve properly, leading to chronic inflammation characteristic of IBD. And it can activate pathways that are implicated in Crohn's disease. Again, microbiome alterations, disrupting the gut microbiome, reducing diversity, and promoting a pro-inflammatory environment. Such dysbiosis resembles the microbial imbalance that is seen in inflammatory bowel disease patients, where beneficial bacteria, for example bifidobacterium, are depleted and opportunistic pathogens thrive. Again, epithelial barrier damage. Salmonella produces toxins and effectors that damage the intestinal epithelium, leading to leaky gut, and this breach allows luminal antigens and bacteria to interact with the immune system, potentially triggering or amplifying the autoimmune responses seen in IBD. Another theory behind this is molecular mimicry. Some researchers hypothesize that Salmonella antigens may resemble our self-antigens in the gut, leading to an autoimmune reaction in predisposed people. Now, this is less well established for Salmonella than other pathogens like Klebsiella, but it is a plausible mechanism. And of course, persistent infection or inflammation. In rare cases, Salmonella may persist in the gut at low levels after acute infection, acting as a continuous inflammatory stimulus. It's been observed in animal models and could contribute to chronic conditions like IBD. And here's some studies. Again, you can come in the database to read that. Here's about genetic susceptibility and differences from irritable bowel syndrome. While post-infectious irritable bowel syndrome involves functional symptoms without structural damage, inflammatory bowel disease features visible inflammation, ulcers, and tissue destruction. Now, if you come down with food poisoning, how do you know if it's Salmonella? Unfortunately, the only way to determine that is through testing from a lab. But you could just figure that it's likely that it is Salmonella and treat it accordingly. Now, because it's difficult to treat with drugs, with antibiotics, some medical alternative treatments have been developed, and that's what this section is about, especially because there's a lot of antimicrobial resistance in Salmonella species, coupled with the growing need to reduce antibiotic usage in both human and veterinary medicine. So we've got bacteriophage therapy, and you can click on that to learn more about what bacteriophages are. It's a very cool type of a virus that kills bacteria. I'm not going to go through all of this, but I invite you again to come in the database and read this, because we want to get onto the herbs here pretty quickly. And then they're also developing antimicrobial peptides, so you could read about that here. And then, of course, we've got a lot of studies that you can come in and learn about. So here is a list of different herbs that have been used successfully. I'm not going to go through all of this again. I want to scoot over to a comparison of herbal treatments and cover that in this video. Alright, so a variety of herbal agents have been investigated for their potential to treat infections caused by Salmonella enterica. And in this section, we're going to compare schizandra, garlic bulb, berberine, lemon, paprika extract, pomegranate peel extract, and andrographis in terms of their antibacterial efficacy, their mechanisms of action, supporting in vitro and in vivo evidence, and possible roles in context of clinical or food safety applications. So we'll start with schizandra. Schizandra fructus just means, it's a pharmacological term that means the fruit of the schizandra. It's demonstrated considerable antibacterial activity against different Salmonella enterica serotypes, with both the aqueous or water and methanolic extracts showing effectiveness. I'm just going to kind of skim through some of this so the video doesn't get too long. Schizandra extract led to lower mortality, reduced bacterial loads in feces, and less clinical and histological damage in organs such as the kidney, liver, intestine, and spleen, suggesting both direct antimicrobial effects and immunomodulatory support, contributing to the control and mitigation of Salmonella infection severity. Importantly, its broad-spectrum activity and clear demonstration in vivo models highlight it as a robust candidate for managing salmonellosis. Now note there it says it's broad spectrum, so even if it's not Salmonella, maybe if it's Clostridia or Shigella, Schizandra might also be used in those cases. So think of Schizandra if you have food poisoning. And then of course our trusty old garlic bulb. It again has broad spectrum antimicrobial properties, and that's probably mediated by its organosulfur compounds, such as allicin. Multiple studies show that garlic extracts are effective against Salmonella enterica, including its common serovars. The antibacterial action is largely bacteriostatic. In other words, it doesn't kill them, it just kind of stops them in their place. They can't do anything. And that's achieved through cell membrane disruption. And it can also impair biofilm formation. Synergistic effects with other natural agents, such as lemon peel, enhance its antibacterial efficacy. Its antimicrobial effects are dependent on both the concentration and the microbial load. While most data from lab experiments results consistently reveal garlic is a potent natural alternative or adjunct to antibiotics against Salmonella, especially in the context of rising antimicrobial resistance. All right, berberine, that's another one of our tiles. It has significant efficacy against Salmonella, supported by in vitro and animal model studies. It's particularly effective at inhibiting the growth and biofilm formation of Salmonella, which is important given biofilm's contribution to antibiotic resistance and persistence of infection. Berberine acts by repressing regulatory genes involved in biofilm and virulence and disrupts the type 1 fimbriae used by Salmonella for adhesion and colonization. Plus, berberine can work synergistically with antibiotics, enhancing bacterial clearance and reducing the required antibiotic dose. In vivo, berberine reduces intestinal bacterial load, alleviates symptoms of salmonellosis, and improves gut morphology and survival rates in animal models. It also modulates host immune defenses by inducing innate immune responses, supporting its potent dual mechanism of direct antibacterial and host-mediated activity. Collectively, berberine stands out for its robust direct and indirect actions and its potential in adjunct therapies for multi-drug resistant Salmonella strains. Now when we come to lemon, this turns out to be most effective in food decontamination. An application of 10% lemon extract to whole and fresh cut produce, such as cucumbers, significantly reduces Salmonella populations after 1 week of storage. Paprika extract. It's increasingly recognized for its broad antibacterial spectrum, including significant in vitro effectiveness against Salmonella enterica. Aqueous and ethanolic extracts have demonstrated strong antimicrobial potency, often comparable to or surpassing certain conventional agents or other spices. Capsaicin and other phytochemicals in paprika contribute to this effect, causing bacterial stress and direct growth inhibition. Animal and food model studies corroborate these findings, indicating that paprika extracts can reduce bacterial populations efficiently. The majority of the data is derived from lab experiments, and clinical or in vivo evidence is limited. Its performance in natural food safety interventions is promising and may have applications in both preventive strategies and as an adjunct to more established treatments. And then we have pomegranate peel. You can make that yourself by tincturing the peel of pomegranate. It's distinguished by its high content of phenolic compounds, flavonoids, and tannins, which confer potent antibacterial activity. Aqueous, methanol, and ethanol extracts have shown significant inhibition of Salmonella enterica, including multi-resistant strains, with inhibition zones often rivaling or surpassing some standard antibiotics. Mechanistically, it disrupts bacterial cell structures, inhibits metabolism, and interferes with biofilm formation, leading to efficient bacterial cell death. In some trials, moreover, the antioxidant and anti-inflammatory properties contribute additional therapeutic benefits, enhancing its appeal as a complementary or alternative treatment. And andrographis. Its primary active compound is andrographolide, and that has attracted attention for its bactericidal, meaning killing bacteria, and immunomodulatory activities against Salmonella. In vitro studies confirm that methanolic and ethanol extracts, as well as pure andrographolide, inhibit various Salmonella serotypes through mechanisms that include cell membrane disruption and suppression of bacterial growth. In animal models, administration of andrographis extract results in reduced bacterial colonization in organs, lower systemic infection markers, and restoration of intestinal health with improved survival rates. These benefits stem in part from andrographis' ability to modulate white blood cells, downregulate pro-inflammatory cytokines, and support overall immune function during infection. Notably, combination therapies using andrographolide with frontline antimicrobials may show enhanced effectiveness and offer potential for treating resistant strains. And so here is an analysis comparing all of these. Pomegranate and schizandra emerge as leading agents with both strong in vitro and in vivo efficacy, broad spectrum of action, and demonstrated ability to reduce bacterial load and protect organ function during Salmonella infection. Berberine is particularly valued for its antibiofilm and resistance modulating properties, with potent synergy in combination therapies and strong animal model results, positioning it as a promising agent against resistant Salmonella. Andrographis stands out for immunomodulatory and direct antibacterial effects, making it useful when host immune support is critical as a direct pathogen elimination. Garlic offers robust in vitro efficacy and well-defined organosulfur compounds with established antimicrobial and synergistic potential, though more in vivo and clinical confirmation is desirable. Lemon and paprika extract are prominent in natural food safety interventions, with moderate to strong efficacy as decontaminants and as part of integrated preservative strategies in food systems. The paprika extract, by the way, is often used in cooking, especially in Hungary, and as a food coloring. And then I'm going to leave it to you to come through the rest of this, making some other comparisons. How do we compare garlic and lemon juice? How do we compare pomegranate and lemon verbena? Andrographis versus berberine? Comparative summary down here. So we've got some tiles that look like they could be very useful in counteracting Salmonella food poisoning.